Hello everyone, welcome back to the class. In our previous class, we had learnt about targeting of mitochondrial proteins, especially to the matrix. Mitochondrial proteins are synthesized by 80S cytosolic as well as 70S matrix ribosome. So, proteins synthesized by cytosolic ribosome are translocated into mitochondria post-translationally. 99% of mitochondrial proteins are encoded by nuclear genes and are synthesized as precursors on cytosolic ribosome. Proteins which are to be imported into the mitochondria have three type of locations, that is outer mitochondrial membrane, intermembrane space, inner mitochondrial membrane, and also matrix. So there are four types of locations present in the mitochondria where proteins are specifically imported. So, for a protein to get imported inside the mitochondria, there are specific mitochondrial target sequence which has attached to the proteins which are designated for the import to the mitochondria. So, this mitochondrial targeting sequence is recognized by the import receptors which are present in the outer membrane. If we have to diagrammatically represent this process, so there is a mitochondrial outer membrane and there is a import receptor present here. The mitochondrial protein which is having the mitochondrial target sequence will bind to this import receptor and will allow this protein inside the mitochondria. So there are two distinct translocation complex, that is Tom complex and Tim complex. Tom complex is present out in the outer membrane. Tim complex is present in the inner membrane. So, Tom complex, as it is present in the outer membrane, they consist of 7 subunits and there is receptors in it. Receptors are Tom 20, Tom 22, Tom 70. Recognize precursor protein and will send or transfer the precursor protein to the central component, that channel forming Tom 40. And there are proteins present in this Tom complex, that is 5, 6 and 7. Tom 5, 6 and 7 are involved in the assembly and dynamics of the Tom complex. Whereas Tim complex, which is present in the inner membrane, are of two types, that is Tim 23 and Tim 22. They transport different classes of protein. Tim 23 is made up of three essential inner membrane proteins, that is Tim 50 plus Tim 17 plus 23. The Tim 50 acts as a receptor and Tim 17 which recruits the motor, Tim 23 which is a channel forming protein. In the same way, Tim 22 is also made up of three inner membrane proteins, that is Tim 18, Tim 22, Tim 54. So let us see the structure now. This is the flow chart. See, this is the image of Tom and Tim. The assembly of Tom complex. If we have to decode this, so the outer mitochondrial membrane has Tom complex, the intermembrane space is having inner mitochondrial membrane, and this is a matrix. space. So this is the placement of different types of complex. Mitochondrial targeting sequence are of two types: N-terminal cleavable matrix targeting sequence, which is known as pre-sequence, and non-cleavable internal targeting sequence. So this is rich in protein, charged positively charged basic amino acid, that is arginine and lysine, and also they have this hydroxyl group containing amino acids serine and threonine, but lack negatively charged acidic residues. These signal sequence direct precursor proteins to the mitochondrial matrix, inner mitochondrial These signal sequence direct precursor protein to the mitochondrial matrix, inner mitochondrial membrane and intermembrane space. This sequence is cleaved once it reaches the matrix. It is removed by mitochondrial processing peptidases that resides within the matrix. The another type of internal targeting sequence is non-cleavable. It is not cleaved. These are present in all outer mitochondrial membrane proteins, intermembrane space and inner mitochondrial membrane proteins. Targeting of mitochondrial proteins into the mitochondria, especially to the matrix. It happens by binding of matrix targeting protein will bind to the Tom complex first. The Tom 20 of this complex recognizes the pre-sequence. There is a pre-sequence, or let me write like this: protein which is having mitochondrial targeting sequence will bind to the Tom complex. The Tom 20 of the Tom complex recognizes this pre-sequence, and it transfers the precursor protein to central receptor Tom 22. This matrix targeting sequence directs proteins through the Tom complex. From this, it is transferred or directs the protein to TIM complex, that is TIM 23, which is the inner complex. After this, the TIM complex is associated with PAM. The full form of PAM is pre-sequence translocase associated motor. So this PAM has several components like MTHSP70 co-chaperones, nucleotide exchange factor and other proteins. So PAM is having MTHSP70 co-chaperones, NEF and other proteins. This MTHSP binds to the translocating polypeptide chain, or in simpler words, it binds to the protein and drives its movement to the matrix. So consider protein gets into Tom complex Tom 20, then it directs to the central receptor Tom 22. After that, Tom 22, the matrix targeting sequence directs the protein through the Tom complex to TIM 23. This TIM 23 is associated with PAM. PAM is having PAM is having MTHSP70, which directs that protein, which binds the translocating protein and drives its movements to the matrix. This is the flow chart of the protein which is being targeting to the matrix. After, when the protein enters inside the matrix, there is another component known as MPP, which is a mitochondrial processing peptidase. It cleaves the MTS, mitochondrial targeting sequence, that is also known as pre-sequence. Many imported proteins are passed on to another chaperone. So there is HSP60 where protein gets into it, which helps the protein to fold properly. So this is the fate.